So are you able to see anything on the screen? Anybody unmute and say?
Yes, sir.
Are you able to see PPT on the screen?
Yes, sir.
Yes, sir.
Okay. So we will just go to a recapitulation of the first class so that we will not jump directly into the fragment-based design. So we were discussing about DeNovo drug design. In that, first we have seen the learning objectives, which covers the fundamental principles you are going to learn and structural strategies, algorithmic overflows. Workflows will be having little bit separate class. We need to take it because this algorithmic workflows, which requires generative AI, where so many terminology is present. As and when it is required, I would like to make that clear point. Separate classes will be taken in this point, but some things is coming in this session also. Evaluation metrics are also there, where we want to find traditional methods and AI made differences. Industry applications, ultimately at the end, you will come to know how it is reducing the time and the cost. Similarly, we are having a learning outcome. You are able to differentiate between the visual screening and the scaffold build up via DeNovo design. We are going to have two strategies, which you are already seeing: growing and linking strategy. So this is based on Bloom taxonomy level 3. That is, you are going to have the first one memorize, the second one is understand, the third one is applying. So where you are going to apply which strategy, that you will be able to know. Then you are going to evaluate different software outcomes so that you can pick up what is the best to you. That is Bloom taxonomy 5. You can explain generative models. They are based on the output what you have got, how it is required to be modified to your requirement. That is understanding you are going to have. And you can critique the designed molecule, whether it is having a synthetic feasibility or not, can we apply in the market or not, you can see. And analyze the one case study so that you will be learning the analysis of this outcome, whether it is suiting to your need or not. So this is the one which you already explained, different stages of drug development and discovery. So the drug discovery part is appearing in the CAD. The development part, what you have got clinical studies and post-marketing surveillance and journey of a drug in the market. So these are all the four which we have already seen. One is Autodeck, where the dock receptor based, DeNovo based, that is grow and listen softwares are used, ligand based PSC are used, computational based, where both receptor and ligand is not known. We are going to use a high throughput screening. Just I am jumping into the topics so that we will be reaching to our own point early. DeNovo, what the meaning is, we are starting from the fresh, and it is from the beginning. We don't have anything in the hand. but we will be starting with structural determination of our receptor. Then we are going to have a modifications of a ligand and make the ultimately the decision whether it is fitting or not. For that purpose, we are going to have two approaches, which one is structure based, another one is a ligand based DeNovo design. And both the structure and ligand based, we have got atom based and fragment based. We can start with the atom and building on atom to make a complete drug molecule, or we can start with a fragments of molecules and link them and join them later to form a drug. So it is having two types of the methods. One is outside in and inside out. In the outside in, we are going to have a specific functional group that bind these connected together and give the real molecule. In the inside out, we see the binding site, and first we will be fitting according to the energy function, and we make the molecule build further. So both methodologies where we are going to use the flexible molecule approach is better when compared to a rigid, because our molecules, they behave in the real time as a flexible. Key steps, these are all the key steps in the DeNovo synthesis we have already seen. We were yesterday at fragment selection and we have stopped. So in that, the primary constraint is we need to have the three-dimensional array of the pocket of a structure of protein. That is the primary constraint. So after the structure of primary structure, it is known and the pocket is known, we have to derive the interaction sites, where we are calling it as a active sites. And we are going to, after having a molecular construction based on what the active site we have got. One is a ligand growing approach and a ligand linking approach. So this is actually what we have arrived at a fragment based design yesterday. We will be continuing now. In between, if you have got any stop sharing, the PPT is not there, just let me know by unmuting yourself and we can correct it. And any doubts are also, you can interrupt in between also. No need to end further at the end. So in the fragment based DeNovo design, we are having two approaches. One is DeNovo linking. So in the DeNovo linking, first we are going to see the active sites. Primary constraint is over, and take the corresponding fragments which are fitting to that particular active site. And later onwards, we will link them with small fragments we are going to take from the library. Then we are going to get a designated compound that is designed perfectly fitting into the pocket. The second approach is growing approach, where we start with one pocket and keep on adding some more fragments. And based on what the interaction site is having affinity, then we can take a suitable fragment and grow the molecule. Ultimately, we are going to design the compound. So in the fragment based DeNovo design, we are arriving at from the scratch only, the molecule which is not existing in the market. We can have both the methods. So the fragments, atoms, or the building blocks are either placed at a key interaction sites. That is actually what we have got a main thing we are doing in the fragment based design. So we know the active site. That active site is having some functional group or hydrogen donor or acceptor lipophilicity. Based on that, we corresponding fragment we choose. Or we are going to take that pocket structure and pre-doc using another program and select a fragment from the database. Then we are going to join together using a predefined rules which are existing as a governing to yield a correct moiety that is going to work in nearby future. The linking of the groups and the linkers may also be predefined according to the feasibility of a synthesis, what we are going to have, and generate the structure which is requiring all requirements are met, and it will satisfy all the conditions. Another approach what we can see in this particular fragment based is a lattice based method. In the lattice based method, the lattice is placed in the binding site based on the atoms around the key interaction sites and are joined using the short part method. Previously, we have seen one atom corresponding to the interaction site. We have fixed it and we are growing it or putting the fragment. In this particular one, we are going to have interaction site and we see what is the shortest path. We can have a join so that the size of the molecule under the control. So then various iterations we have to do it, each of which include the translation and rotation or mutation of the atoms. So shifting, we are going to have isomers also, and we are having a spatial recognitions where we need to do some more modifications. They are guided by the potential energy function, eventually leading to a target molecule. So this lattice method, either it can be having a fragment fixed and making a short path to have a linking, or we are going to start with one and making a short path to the next site, and we are going to join them to get a new molecule. Another approach is the molecular dynamic methods. This is completely the expectation what we are going to have in the real-time scenario, where the molecule is not fixed, it is going to move. So the building blocks are initially randomly placed, and then by the molecular simulations, they are allowed to rearrange. And we will be finding in particular direction, the molecule or the fragment, what we have attached in the interaction site, it is going to work and having a proper binding. After each rearrangement, certain bonds were broken and process repeated so that what we are going to have a perfect fit. So during this procedure, each time we are going to generate a scores. So the score is generated for each structure, what we have done in the modification in the fragment rotations, and with that will be stored for the evaluation. And the best score, whatever the structure which is having, we will be having for the Pareto principle, and we will pick up the best one, and we are going to go further. So in this journey, what we have done is the first we identified the interaction site. So first of all, atoms are present in the binding site are analyzed to identify those that can be part in the hydrogen bonding interaction. So, and also we can see what are the part which can have a van der Waals interaction also. Most of the times oxygen atom and tertiary nitrogen atoms are identified as a hydrogen bond acceptors. That is already present in the receptor interaction site. We have to identify that. So if a hydrogen is attached with the nitrogen or oxygen, it means that there is a hydrogen bonding interaction is present. The corresponding group we are going to pick up from the database in the fragment selection. So the aromatic and aliphatic carbons are identified because they are capable of taking part in the van der Waals interaction. The same principle we are going to use for the linkers also, what the fragments are linked later. So this can be done by the aromatic or aliphatic carbons. We are going to have a linking. So the interaction site, these are the position sites that define where the ligand atom can be placed to interact with any one of the above atom present in the that is hot spot of a receptor. For the X bonding site containing methyl group, program will be identifying the carbon of that group as a aliphatic carbon capable of taking part in the van der Waals interactions. So like that, we are going to have each bond and each atom which is present there that is actively taking part in the binding that we are going to consider for the selection of the fragments and linkers. Then we are going to fit the molecular fragments. So once the interaction sites have been determined, which is with the proton donors and acceptors, the LUDI program, LUDI program, that access the library of the several hundreds of the molecular fragments present in the database, and it matches with the what the interaction site which is already present. Generally, what we need to put a upper and lower limit in this particular journey is it is not worth to do so many atoms present in the fragment. Otherwise, what will happen? We are going to lose some of the features. That is the reason the molecules chosen are typically 5 to 30 atom in size, 30. So the molecules chosen typically the 5 to 30 atoms in the size. They are usually rigid structures, because the fragment we require to be rigid, then linkers it will take the flexibility for the rotation. So because of the fitting procedure, assuming that the rigid fragments, the fragments have a different conformations also, because when we are rotating in 360 angle, the fragment, how it is behaving with interaction site, we need to identify and we need to put that into the account for our further optimization. So there is no number of fragments are present in the library and the database of the library. If they are to be represented by fairly by the fitting process, each conformation is treated as a separate entity during the fitting process. Because in this particular journey, sometimes we will be getting cis isomers, trans isomers. We are going to asymmetric atoms which are having dextro and levo rotarity. That is going to be a perfect fit there. So such conformation only we have to select further, so that our drug-like molecule is coming to the near reality. So the fragment is fitted to the binding site due to its interaction. And say, for example, if you take a acetone fragment, it is aliphatic in nature. So it is fitted onto the aliphatic interaction sites. So corresponding site it will be selected based on the what the interaction site nature is. Carbonyl group is defined as the hydrogen bond acceptor, which can be fitted into the hydrogen bond interaction sites. So CO group, where it is present in the fragment, which can be attached in the interaction site. Like this, most of the times the computer itself pick up these fragments for the interaction site and suggest for you, and it will give the scores. So the fragment bridging, after the fragment selection is over, we need to bridge the fragments. Once the fragments are identified, then are fitted into the binding site. The final stage is to link them up in the fragment based DeNovo drug design. So the program first identify the molecular fragments that are closer to each other in the binding site, then identifies the closer hydrogen atoms. So that is going to have an interaction little bit later, that is going to create a bonding. So that is interaction is going to be perfect and it will work as a drug. The program now tried to the various molecular bridges are stored in the library to find out one program which is generally being used as a sprout. So another one is SPROUT and ligand. So don't get confused with the software names and the database names. Later onwards, we will, once the session is over, you can make easily recognition of each structure and each database, how it is can be done by the software. Next, once we are making a linking and the fragment selection and the linking, we need to have a secondary target constraint. So this is binding affinity alone does not suffice to make the effective drug molecule, because we have seen till now fragments and linkers are selected in such a way that they are having a good affinity in the binding site. But it is not alone sufficient, because once the molecule is made, it has to be given to the human. It is having absorption, distribution, metabolism, excretion, and toxicity. So this we entire together we call it as a admit it has to pass. So it should be first absorbed, then distributed and optimally metabolized and excreted without having a toxicity. In general, most of the time to meet these requirements of ADMET, we are having some rules, what we have already seen, Lipinski rule of 5. That is, we are reiterating here. First session, we have got second question in this, third question. So we are having to repeatedly the same thing will come, because most of the molecule meet this rule. Now we are entering into the rule-based approach. Of course, it is traditionally present. Here, the first rule, we know that not more than 5 hydrogen bond donors, that is OH and NH groups, and not more than 10 hydrogen bond acceptors, notably nitrogen and oxygen. Now we are getting confused between these 2 points. We will like to little bit deeper into that, because it is having these 5, 10, whether it is acceptor or donor, that confusion will be arising from starting also. So in the next slide, we'll see how to make this difference. The third rule is that molecular weight under 500 Daltons. And the fourth rule is log p value, which is less than 5. That is the ratio of the concentration of the solute in the solvent. That is when compared to your octanol and water. Log partition coefficient value, that should be log p value under the 5. Under these rules, as a filter, the resulting compounds are more likely to have a biological activity. Previously, we have seen the perfect fitting. By following these rules, they are passing these biological activity constraints also. Then we can work that particular moiety. Now, each group, how they are going to be considered, whether the group is having a proton donor, that is hydrogen bond donor, or acceptor. So most of the times these group will work both the manner. That's what we need to see, because this is a little bit confusing to us whether we consider that group as a donor or acceptor, hydrogen bond donor or acceptor. Most of the molecule, they possess both the activities also. So the alcohol, say that is OH group present in the ethanol, it will be acting as a hydrogen bond donor also, hydrogen bond acceptor also, because it is containing donor H and acceptor O, that is oxygen. Similarly, primary amine, NH2, say for example, it is in the methylamine, it will be acting as a donor also, because it is containing 2 hydrogens, and it will act as a acceptor also, because it is containing the nitrogen. Similarly, we are having a ketone, so that is present in the acetone group. It is not a donor because it is not containing any hydrogen atom, but it can act as an acceptor. I think now you are able to understand how to make this oxygen and nitrogen atoms, whether they are donors and acceptors, how to make them and see them when we are taking the rules. Another one is the ether. So diethyl ether, you are having a ethyl group on either side of the oxygen, where it is not having a donor capacity, but it is having an acceptor capacity because it is having a oxygen atom. Tertiary amine, it is having no donor capacity because there is no hydrogen, but it is acting as an acceptor. You see in the second row, you have got a primary amine. It is acting as a both donor and acceptor. But the tertiary amine, which is acting as only acceptor, not the donor. It means the loci of the atoms which are present, their ability to donate hydrogen or accept it, in all the cases, they are accepting it. They are acting as acceptors. So, and the donors will be very less. And go back to the rules. Not more than 10 hydrogen bond acceptors. It is the rule which is present. That is 5 into 2. That is the rule of Lipinski rule of 5. Notably, nitrogen and oxygen atoms, it is been clearly given. And the first rule is not more than 5 hydrogen bond donors. There is also we are having OH and NH groups. So based on this, we can see that each the fragment and the linker, how many acceptors are present, how many donors are present, we need to add them. And we need to see that whether the biological activity and the ADMET rules are fulfilled or not. Once we confirm it, we can go further. We need not do manually, but we can do manually also by seeing these atoms in the molecule what we have generated, but software will take care of it and it will give the score. Next, coming to the log P values. So we are having one concept is the molecular weight less than 100, because we are taking the fragment which is in the range of 5 to 30 atoms. So it will fit generally less than 500 only. We have seen the hydrogen donor, that is hydrogen bond acceptor, and the donors we have already seen. Now the fourth rule is summary of the effect of log P. So the log P values, how these groups specifically affect the log P value, that is lipophilicity of the drug candidate. Lipophilicity should not be too high, and also it should not be too low. So we have got one trick is called as IMHB trick. So that is trick is used to rising the log P values internally. So this how it is going to be done. We will see it. It is a clever strategy in the DeNovo design. It is the use of intramolecular hydrogen bonds. Intramolecular hydrogen bond means, say for example, you have got one molecule which we made based on the fragments at the different interaction site and linkers. They are containing hydrogen bond donors also, acceptors also. So they can attach internally with the atom of own molecule itself. That is called as IMHB. So this occurs when donor and acceptor within the same molecule bond with each other. When they are having a pocket big, we are having a linkers. So we are going to have short pot method also. In all the cases, we tend to have these hydrogen atoms also, hydrogen bond acceptors also. In those cases, whether it is having a IMHB or not, that is intramolecular hydrogen bonding or not, we need to see. Just you see these particular summary effects of log P. So the modification, what we are going to do and what is its effect. If you see it, then you can understand the concept very well. That is what we have got a Bloom's taxonomy level 2. Say you are adding OH or NH2 into the particular group, it decreases the log P value. How? Because it is having a increasing the hydrogen bond donors and acceptor, molecule stays in the water. That is the reason it decreases the log P value. Because it is having a both hydrogen bond donor and acceptor. So in the hydroxyl group, it is having one hydrogen and acceptor one oxygen. In the case of two hydrogens, in the case of primary amine and one acceptor N, that is nitrogen. Similarly, by adding CO or O, it decreases the log P value, increases the hydrogen bond acceptor. That is the reason molecule stays in the water. Similarly, by adding halogens, that is chlorine and fluorine, it increases the log P value. It adds the hydrophobic bulk with the minimal hydrogen bonding capacity. That is the reason log P value will be increased. Similarly, by forming IMHB, that is the intramolecular hydrogen bonding, within the molecule itself, the hydrogen bonding is occurring, not with the interaction site or active sites in the proteins. What will happen? It increases the log P value. Why? The polar groups present within the molecule, they cancel each other, they bond with each other, that will not be available for interaction with the interaction site in the protein. That is the reason the log P value will be increasing, water solubility will be decreasing, because they are getting a nullifying effect. So these rules which will understand when we are selecting a fragments are linkers in the fragment based design, so that we will be arriving at a correct combination so that it is fitting perfectly and following the Lipinski rule of 5. This is what the strategy in the fragment based design we need to adapt. Now, sir, I am having a problem in the selection of nitrogen and oxygen and corresponding hydrogen bondings. So can't we use bioisosteres? The bioisosteres not like isomers, what we have seen, but they will be resembling with respect to the electronic effects and the steric effects with the same as that of the original molecules, that is fragments or the linkers which are present with the oxygen or nitrogen. So we see generally what the major effects of it, so that we can select the bioisosteres, because you are having one 2 marks question in the bioisosterism. That is going to have the helping of what the molecule which is having a impact onto the log P values. So the bioisosteres in this particular case, say for example, if you are replacing, that is original group is carboxylic acid, that is COOH. If you are replacing with the tetrazole, then it increases the log P value. So because it is the tetrazoles having a 10-fold more lipophilic than the carboxylates, that is the reason it increases the log P value. So sometimes where you are having the constraint in the secondary constraint is dominating to make a drug-like molecule, so we can use this bioisoster strategy in the fragment-based design. Similarly, if you are replacing carboxylic acid with acyl sulfonylamides, that increases the log P value. Similarly, cyclic sulfonamides will also increase the log P values in the place of carboxylic acid. If you want to have a log P value manageability with the corresponding bioisosteres. Similarly, you can replace hydroxyl group OH with fluorine. That will increase the log P value because it is a smaller in size, but it is hydrophobic in nature and it is not having a hydrogen bond donor effect when you replace OH with fluorine. Thiol groups in place of O, we are replacing with sulfur. So thiol group increases the log P value because the sulfur is larger and less electronegative and polar than the electronegative or polar than the oxygen. That is the reason it is having increasing effect. Amide group, if it is replaced with triazole, it increases again. And trifluoromethyl group, that is in place of hydrogen, we are using a fluorine. That is going to have a more hydrophobicity and less proton donating capacity. That is going to increase the log P value. Now you see the phenyl rings, if it is with bicyclopentane ring, that is called as a cubane, that decreases the log P value. So this saturated cages, which are like a bicyclopentane BCP, that is also called as cubane, provide the similar geometry with as that of the phenyl ring, but improved solubility because it is having a decreasing the log P values. Whenever we are selecting these log P values with respect to the linkers in the fragment-based design, if you are seeing these primary effects on the log P value, we can select replace a better linker there, or later functional groups are the fragments in the drug moiety. These rules are already trained in the software, what we have already seen in the Sprout and Legend, where you are going to get picked by the software in a reasonably correct precision, and we can get the correct moiety without any problem, because we are going to get ultimate scores also at the end. Then we are going to have a receptor enzyme interaction and its analysis. We have selected and we have seen the rules of 5, and we have selected the fragments and linkers, and we need to see the interaction and its analysis. So the ability of the protein receptor to bind or interact with the ligand depending upon the formation of the weak non-covalent bond between them, and this process release on the sequence of the amino acids in each protein in the way in which the side chain interact with each other, whether it is electrostatic interaction, hydrogen bondings, weak interactions, non-covalent interactions. Based on that, we are going to have a binding capacity. Most of the times, these bonds, it will be having a glue to the molecule which is attached to the protein. Just like a glue, we are attaching paper onto the wall like that. It is acting as a glue. Whereas this lipophilicity values, it will act as a wax. It lubricates and smoothly goes into the pocket. That is the reason both these hydrogen bond donors and acceptors and your log P values are considered with great care. Ultimately, we need to fit the size. That is the reason molecular weight less than 500, we have already taken as one rule, so that it will be fitting into the pocket easily. So in this particular journey, different side chains can form the different bonds because of the protein interaction. It can be very perfect basic of the, and this is actually the specifically we are going to have, because the different side chains which is having a different bonds because of this protein interaction. This is also specific with respect to the hydrogen bond donor and acceptors. Based on this phenomena, we are going to have a molecule which is coming out as an agonist or also antagonistic. So because we are having a DeNovo synthesis, we are going to end up with a molecule which is not existing previously. So here, whether it is agonist or antagonist, we want to see now in what way the molecule what we generated will act as agonist or it is going to act as antagonist. We need to find out now. So the endogenous ligand, which is already present and it is interacting with the receptor, when the endogenous ligand like the insulin is present, it is having an interaction with the insulin receptor, that is going to cause a inflow of your glucose, resulting into a conversion of ADP to ATP and releasing the energy. So this is actually what the endogenous ligand bind to the receptor and produce the effect. So this is present already in our body, it is existing. In the case of the drug is an agonist and receptor, so the agonist have the similar shape of that particular endogenous ligand what we are using. So that is already present in the nature. So what we are using as a drug is having a similar agonist shape. So that is the reason. So they attach to the endogenous ligand, so bind to the receptor and produce the same effect as that of the endogenous ligand. Then we will have an agonistic effect. Because the endogenous ligand and what we have produced the drug ligand, both are having a similar shape. It is producing a similar effect. That is the reason we are calling it as a agonist. Whereas the antagonist and the receptor, the protein, there is no effect shown by the antagonist because the drug is having closely related shape of the endogenous ligand, but it is not having the interaction as that of the ligand and block the binding site, not reaching the original endogenous ligand to reach there and stop the ligand to bind. That is called as a competitive antagonistic attachment in the pocket. That will be acting against it. This will be acting as a antagonist. So here, whether the shape is having the same, it does not mean that it is going to work as a drug. It may act sometime as a antagonist also, because it is having a closely related to the shape of the endogenous ligand, but it is having a blocking effect. Because of that blocking effect, the endogenous ligand will not attach to the receptor. Because of that reason, you are going to devoid of any effect and you will not produce any biological effect which we are expecting it. So it is acting as a antagonist. So in this particular fragment-based DeNovo drug design, this feature is very, very important. We need to consider. Otherwise, the drug what we are seeing now is resulting into another category of the drug which is opposite to disease what we are searching for. So what are the theories of this particular interaction? If you know it, whether we can have a agonistic effect or antagonistic effect, we can find out. This is very, very important and we need to consider. Here, before going to the session, can anybody is having a doubts or anything, we can unmute it or ask any question. Otherwise, we will proceed further. As of now, 39 are there. It is a good number and it is an important question also. So that we will be having the same thing which is in the term end also, and your sessional also we are having it. And it is going to meet all our Bloom taxonomy level and learning objectives very closely syncing to our requirement. Okay, done. We will go the theories for the drug receptor interaction. Over the years, a number of hypotheses have been proposed before the intervention of the computer into the account in our research. So the ability of the drug to interaction with the receptor, they elicit the biological response. The first theory which is proposed was the occupational theory. It is proposed by the Clark. It states that the intensity of the pharmacological effect is directly proportional to the number of receptors occupied by the drug. Say, for example, we have got a receptor availability 1 lakh. If all are getting attached, occupied with the drug, then it is having a directly proportional effect. After they are having a attachment to all receptor, then there is no further. It will be having a plateau effect, plateau stage, and there will not be any directly proportional relationship. There will not be any because the receptor got saturated. It means till the receptors are getting saturated, the occupational theory, it is having it says that a directly proportional effect to the number of the receptors present occupied by the drug. So the L is a ligand plus a receptor. It will be forming as a bond that is complex receptor and the ligand complex. And it is having a irreversible reaction. One is association and another one is a dissociation. The complex of the protein and the drug, they are getting dissociated to form as a free moieties. And again, they will be associated to form a complex again. This journey, it is producing a stimulus and treating a disease. This is what the classical receptor occupancy theory is. So now, the second effect is called as the induced fit theory effect. What is induced fit theory? We know that in the crystallography and your NMR, whatever we have created to see the internal structure of your protein to find the primary constraint and a secondary constraint. So we have taken as a pocket that is actually not fixed or a rigid. The moiety what we are giving as a drug can change and alter the protein socket or the receptor pocket what we are considering as activity site that will be altered by the drug. That is actually induced by the drug alone or by the environment or the simultaneous diseases. It will cause a induced fit theory. It is given by the Koshland, originally proposed for the action of the substrate with the enzyme. Then it is given for the extended to the ligand and the receptor. According to this theory, the receptor not to be need to appropriate conformation to bind the drug. No need to have a exact shape as that of what the receptor is having. No need. It is having. It is not required. It is the point where you will lose the confidence with the card. What we have done till now, what the journey, what we made as a fragment-based approach is going to fail in this particular one, because when the receptor, what we have identified the steric effects and the electronic effects, what we have seen and considered one fragment-based linkers, and we have designed the moiety. And this induced fit series says that no need to have a appropriate conformation to bind it. It is not required. It means whatever we have thought is appearing to be failed and futile. But it is always like that. It is not like that. That is the reason what we have studied is worthwhile considering. But we cannot rely on that only. Because of this particular induced fit theory, our drug, what we are considering may not work. And sometimes what the drug we have rejected because of it is not fitting into the pocket may work because of this induced fit theory. It means we are going to have at this particular stage false positives also and true negatives also. So we are going to take a positive drug, we reject it. Similarly, we take a drug, we consider it a drug, but it will be failed as a drug. In both the cases, we are losers. So this is a very strange theory, induced fit theory. It is challenging our pharmacist and research community to handle this very carefully. What we are taking assumptions and the predictions will not work in this particular theory. So we are going to lose all our money, what we have based on if we are not considering this particular theory. So this is as the drug approaches the receptor according to the theory, the conformation change is induced in the protein. So the protein, it changes its shape, which orients essential to the binding sites, which is going to fit to the drug. So according to the drug what we have given, the protein will adjust now. That is called as a induced fit. So the fit is induced. It is not actually fit to place it in a pocket, but it is induced because of that reason. Because of that conformation change in the receptor, it is responsible for the biological response. So your drug is acting even though it is not having a perfect fit as that of your pocket, which is present according to our crystallography studies of protein receptor complex. What we have taken and what we have rejected as a drug, but it is working now. So according to this theory, an agonist would induce conformational change in the receptor, and antagonist would bind without conformational change. It is going to, without any conformational change, it will bind and it will block the original endogenous ligand to enter. This is the one which is acting as an antagonist according to this particular theory. So the partial agonist which cause the partial conformational change. So it is actually vague by describing it in the form of a text. If you see the figures, you can easily understand while explaining 10 pages of the district. The bottom you see, you will easily get the point here. First, you see the enzyme, which is having like a triangular active sites, which are having a spaces, the steric spaces, which are having like a triangular in nature, where the substrate, what we are taking as a ligand, which is having one is a square type and another is a triangular in nature. That is actually what is appearing that it will not fit into the work, it will not act as a drug. That is our assumption. But according to the induced fit theory, the substrate, what we are going to give as a square and triangle, it will fit into the enzyme, and the enzyme that is, or the receptor, it will take the shape of ligand. So this is what is happening, and the enzyme substrate complex, which is having same as that of what you have got a ligand. So this complex enzyme product complex will be coming, and once this complex is dissociated after association, the enzyme will take the shape of the reversed conformation. What is originally having, it is got reversed to the new conformation that is present in this induced fit. This is what the challenging to our whole journey. That is reason our pharmacist job is so critical. We need to have a logical approach, that is rational approach, when we are inventing a new drug introduced into the market. And this is where so many drugs, what we consider them as a drugs, will fail, and where so many molecules we rejected them may pass as a drug. This is actually what is making a clumsy situation and challenging. We need to take it as a granted to accept this induced fit theory without any hesitation. This is actually what the reality is. This is where the DeNovo drug design, our drug research is having a challenging to accept, but at the same time, we need to take it to the consideration and proceed further. The third theory, which is having in the interaction, is the rate theory. In the rate theory, the response is directly proportional to the rate of drug complex formation. When both the molecules are having a corresponding opposite charges, then they will have a highest rate of formation. So there will not be any repulsion. They will be having so much of attraction. So complex is formed with each. So this is actually what the rate theory is proposed. So the first theory is occupational, second is induced fit, the third theory is rate theory. So what it says, the effect of drug molecule is based on their rates of association and dissociation of the drugs. So how it is we are knowing this? Because we are having most of the times in our chemical synthesis, we are starting with the reactants and ending with the products. In our drug design, we are starting with a ligand and a receptor and ending with a ligand receptor complex. This is not unidirectional. It is bidirectional. It will proceed to and fro. So the to and fro where it is having association and again coming back to dissociation to original. So this is where the rates are going to work as a primary phenomena and deciding factor. According to the rate theory, pharmacological activity is fundamental of rate of association and dissociation of the drug with respect not to the number of occupied receptors. So we have already seen in the occupancy theory, all are occupied. There is a directly proportional elopement when the receptor is still got saturated. But rate theory says that, my dear, it is not like that. It is a rate it is deciding. It is not that how many receptors are vacant to get occupied with and you don't consider. So it may come to plateau, appear to be pseudo orders, like a pseudo first order, and it will appear like it because of the rate is a dominating phenomena there. So it is so confusing now. So in the case of agonist, what the rate theory says, the rate of both association and dissociation will be fast. They are acting as agonists. In the case of antagonists, association is fast, but dissociation is slow. So the dissociation is slow. That is the reason the antagonist is sitting in the place of pocket. It is not allowing endogenous ligand to attach there. That is the reason it is acting as an antagonist. So in the association and dissociation, both are fast, where we are having the agonistic effect. Whereas partial agonistic effect is having a drug intermediate complex dissociation rates. Like first case and second case, it is having intermediate, then it is having a partial agonistic. So the three categories of the drug now we are having according to the rate theory. One is agonistic, another is antagonistic, another is a partial agonistic. because of the rate. It is not due to the occupancy. It is not the induced effect. It is affinity to bind with a complex, form of complex, where the ligand and your drug is getting attached. So these three theories are very, very important to be considered in our drug design. Not only in de novo synthesis, it is a basic drug design, where we are having a particularly all the three theories are accommodated in our high throughput screening, and what we are making a regular animal studies from trial and error methods, what we have taken into the consideration previously in the age-old phenomena, where they don't see any which theory is following, whether the drug is working or not when you are giving to the animal. That's what they see it. In those cases, which rate theory is not the bother, whether the drug is acting or not, it is a bother. So once it is showing the response, we go ahead as a drug, then we take the SAR studies and QSAR studies. Further, we improve the response. That is the journey which is going in the lab approach. That is using a synthesis of the compound, giving to the screening in the animals. Then we are finding is a drugability. Then we are entering into the clinical trials. But now we are doing reverse phenomena. We don't enter into the lab and we don't go to the synthesis and the animal studies. Just we see in the computer. So computer will take care of all this entire journey and take a molecule. Then we synthesize for the confirmation in the lab and testing in the animals at the last stage. So in the traditional method, we are doing all these things and wasting our money. Now we are taking only one moiety, we are taking to the last, and we are proceeding with that testing whether it is working or not. So the success rates in both the case will be same, but we are going to have some of the speed more with the CAD. That is the reason we need to consider this computer-aided drug design as a pivotal role for managing our drug design and development. Next, we will see in the next class, what are the complex, what the bonds which are required to be used, and what type of the bonds which are having in that receptor drug receptor complexation, that we will see in the next class. I will open the floor for the questions, anything. So anybody can unmute yourself and you can ask the questions.